Awesome. Well, thank you guys all for joining us for the EEMB seminar series. This week, as you know, we have Dr. Alexa Fredston, who is our esteemed guest. She's a former UCSB Bren School PhD student, and she graduated in early 2020 from Dr. Gaines's lab and Dr. Ben Halpern's lab. And while she was at UCSB, she conducted her research and wrote her dissertation on the phenomenon of species range edge shifts in light of warming ocean conditions from climate change. And she's now migrated herself back to the East Coast, where she's a postdoc in Dr. Malin Pinsky's lab at Rutgers University in New Jersey. She's not only a former UCSB student, but she's a friend of mine. Since 2019, I've been meeting with Alexa and a handful of other PhD students, all at kind of similar-ish stages in their graduate school trajectories for a weekly dissertation club. In this meeting, I learned how to write an actually tenable to-do list. Just as a note, before this club, my to-do list would usually have single line items like draft dissertation on them. And I learned quickly that that was not actually a good to-do list. How to hold myself and other friends to our to-do lists and to support each other in those endeavors, and when it was appropriate to serve as a wildly enthusiastic cheerleader. The answer there, which everyone should make a note of, is all the time. And I can genuinely say that Alexa has played a very pivotal part in my development as a scientist, and for that I'm very grateful. So this is partly why I'm so excited to hear about her work today, though the simple coolness and the timeliness of her current work using data science and modeling to study how climate change impacts species range shifts in the ocean, among other important things, is more than enough. I'm really excited to hear about the early career advances that Alexa has made since we were meeting weekly for Dissertation Club, and to showcase those advances to our peers in EEMB. So with that, I will let Dr. Fredston take it away.
Thank you so much. It's a very kind introduction, and I certainly don't think I would have graduated without our dissertation group. It's also particularly special for me to be speaking to UCSB EEMB because my defense was in the MSI auditorium, so I feel like I'm spiritually back there with all of you, even though, of course, we're all at home today. We're going to be talking about understanding and predicting Anthropocene range dynamics in the sea. The core question that I work on is, why are species found where they are and not elsewhere? Why would any species have an equatorward range boundary between the subtropics and the tropics on land in a region with fairly continuous and consistent climate? Why are there some large cosmopolitan marine species like sharks and whales that occupy almost the global oceans, yet their juveniles are confined to relatively specific parts of the coastal seas? How are biogeographic boundaries maintained on land and in the sea? The partner question, I think, to the one I've just posed is, what makes species ranges move? Species ranges are very stochastic in general, they just move a lot. But at the moment, they're also moving directionally. Species are on the move. Around the globe, species are shifting upwards in elevation deeper into the sea and generally towards the poles. This global reorganization of biodiversity is one of the most profound effects of human activities on nature and has serious consequences for nature and for people. There may be some species that actually benefit from climate-related range shifts if new habitat opens up that wasn't previously available. But many others will probably suffer as their historic habitats become too warm. And both of those outcomes can have cascading effects on ecosystems if the species in question play a pivotal role in a community or an ecosystem. Not only do range shifts challenge species and ecosystems, they challenge us, because often the way that we approach natural resource management relies on fixed spatial units and wasn't really designed with the possibility of species moving through them. There are lots of other ways that range shifts affect human welfare and well-being, way too many to list here. One is that climate-related range shifts include disease vectors like mosquitoes carrying diseases further and further away from the tropics than ever before. And they've also affected everything from systems like kelp forests and coral reefs that a lot of coastal communities rely on, and crops like coffee. I'm not going to talk about this much more after this, but I should also point out that if this wasn't enough bad news, range shifts also themselves can have negative feedbacks with climate. In particular, some range shifts, for example, let's say the range shift of bark beetles in North America, one that a lot of us are familiar with, are actually exacerbating climate change. As bark beetles have spread throughout North America, they've killed a lot of trees. That both reduces the carbon they can store and makes them more prone to fire, both worsening climate change. So I'm not going to spend any more time convincing you that species are shifting their ranges because of climate change. We know it's happening. It's been documented around the world. The challenge, though, is that although we're quite certain this is happening, climate alone doesn't explain the distributions of species. That paradox is really what I work on, how to understand species shifting in response to climate, but recognizing that we need to look at more than climate to get a handle on what's going on. Let me give you a few examples of what that looks like. Here are just a few figures from papers from all types of species. The top left one is showing the latitudinal distribution of one particular bird species that the authors chose as an example. And the two colors represent an early time period that was cooler and a late time period that was much warmer. I didn't even label them because you can see their distributions are basically identical. So we would have expected this bird species to shift north because of climate change, and instead it didn't move at all. You see something similar in the bottom left plot. This is now North American forest inventory data looking at trees. And the authors asked what had happened to the northern range boundary here. A positive value would be an extension towards the poles. And the southern range boundary here, where a positive value is an extension toward the tropics. So what they expected to find is to have tree species largely be here, that they're kind of marching northward, the northward boundary is extending and the southward one is contracting. Instead, as you can see, these percentages show the number of species. They did find a few species that fell in that category, but they also found almost as many that were marching south, a very small handful that expanded their ranges, and the most common thing was just that ranges were shrinking with basically no relationship with climate. The circles here just represent seed size of the trees they studied. And I'll give you a marine example. This is going to be a mostly marine talk. The plot on the right is about 20 plankton species that the authors modeled with different classes of statistical species distribution models. And their goal was to beat what's called a persistence forecast. That's why it says no difference is the SDM compared to the persistence forecast up here. It's a relative plot. A persistence forecast just says, we don't have any idea what's happened to this distribution. So we're just saying the range has been the same since our last year of data, no change. For a species distribution model to have beaten that for plankton, the bar on this right-hand plot would be green. As you can see, there's not very much green here. So as ecologists, you probably are already thinking of explanations for these plots, why these authors got these results. And it really shouldn't surprise us that ranges are not perfectly and uniformly linked to climate, because there are so many other processes that we know really matter in governing species distributions. This is an incomplete list. Some of these probably played a big role in the plots that I just showed you. We're going to talk about a number of these today. I'm interested in all of them and happy to talk about all of them if it's what you work on. So we'll dig a little bit into temperature extremes, how they limit or don't limit ranges in the oceans, dispersal limitation. We'll talk about oxygen, its role in the oceans a little bit, and then we'll also go into population dynamics. Some of these other processes, like I said, hopefully some of you are studying and we can talk about them. So the two projects that I'm going to go over today, one is an empirical test of range limiting mechanisms focused on temperature tracking at marine range edges. And the second one is on my postdoc project, which is building a process-based model of species distributions to create short-term forecasts for 4 focal species of importance to commercial fisheries in the mid-Atlantic. Before we get really into it, I just want to quickly be sure we're on the same page about some definitions. Today we're talking about ranges, which is the geographical extent of a species on the planet. The way we conceptualize a range can be very focused, like this range map for giraffes, where you can see that clearly they have relatively precise information about where giraffes are found in Africa. And if you went to one of these orange patches, you might be relatively likely to see a giraffe. Contrast that with the bottom map for bottlenose dolphins, which basically says they're almost everywhere except incredibly cold water. And I don't think it's super likely that if you went to one of these points randomly and looked down, you would find a bottlenose dolphin. So just keep in mind as we go forward that the definition of a range can vary a lot based on who is measuring it and how much data you have. Of course, when a range moves, that's called a range shift. And we think about range shifts unfolding sort of at 2 frontiers. One, and we kind of hinted at this with the tree plot. One of them is the poleward edge, the one facing the poles in the northern hemisphere, it's north. And people also call it the cold edge because it faces the colder part of the range, or the leading edge if the species is shifting the way we think it will. I try not to use those words because we don't always know if it's shifting the way we think it will. And at the other edge, we have the equatorward or warm or trailing edge. I also want to acknowledge the data that I use before I really dig into the studies that we're going to go through together today. All of it is from the National Oceanic and Atmospheric Administration, which since the 1960s in most US waters has gone fishing every year and recorded what they found. This is a massive effort. It's one of the largest spatial extent data collection programs in the world. And it requires just a tremendous amount of knowledge and effort. So I study biodiversity, I study climate, but I never actually leave my desk. It's even more true now. And it's only possible because of NOAA staff members who've been collecting this data since long before I was born. So I really can't say how important this is to my ability to study biodiversity and global change. We're going to start out by focusing on range edges, which is kind of where the rubber meets the biogeographical road, in that that's the point beyond which a species doesn't persist. It doesn't have stable populations. And so we know that it represents some limit on where that species can live. There's another reason to study range edges, though, which is that I think it's often what's most important to people who are managing changing ecosystems and trying to deal with climate change. As ecologists, we often get asked things like, when will a certain species turn up in our region, or when will another species vanish? And those are questions about range edges. Despite that, range edges actually haven't been studied that much in a time series way in the context of climate change. And that's because it's a little bit challenging to actually do it. There's no universal definition of how to measure an edge. They're very hard to track through space because edges can be very variable. So you need to have data collection over a massive area to be sure you're really capturing the dynamics of the edge. And we also don't really have a universal theory in biogeography of what should be limiting a range edge. There are a lot of different hypotheses. Sometimes they conflict. It's hard to know what you expect, and that makes it difficult to design effective tests. The first test that we did was comparing two prominent theories of range edges in biogeography. The first is one that's universally thought to be pretty prevalent in the oceans, particularly with marine ectotherms, which I call the thermal niche hypothesis. This basically says range edges are temperature mediated. When temperature changes, the edge will move. This is based on the fact that a lot of marine ectotherms have geographical distributions that line up really well with their thermal limits as measured in the lab. And that suggests that their geographical extent is really limited by a physiological constraint about temperature. So as soon as it gets too hot at the equatorward edge, they have to move. And as soon as it gets warm enough for them to spread beyond their cold edge, they will. There's another theory that we also tested that comes from the biogeography literature, and mostly terrestrial literature, that is called the species interactions abiotic stress hypothesis. This basically says, okay, the poleward edge, sure, is limited by temperature. But at the equatorward edge, where you have less temperature variability and more biodiversity because it's the edge facing the tropics, species are likely to be limited by competition or other interactions. Of course, these aren't the only theories of range edges, but this was our point of departure for starting to really test what's going on with range edge dynamics in the oceans. We started this project in the Northeast US because it has the longest NOAA survey time series and the most consistent history of warming. The first step, which I won't show you, but I'm happy to talk about, was to delineate which species actually have a range edge in the region. That's non-trivial because marine species have enormous ranges. And so we had to use multiple methods to kind of convince ourselves that either the poleward or the equatorward edge actually fell somewhere in between Maine and Cape Hatteras so that we could capture its dynamics. I should note here that for all the range edge stuff I'm about to present, we don't have both the poleward and the equatorward edge of the same species because the ranges are just so huge. If a species has a poleward edge here, its equatorward edge is like somewhere near the equator or the tropics. And we just don't have any data sets that are that large. So when we're talking about poleward and equatorward edge dynamics, they're not necessarily the same species. But we quantified them as percentiles of latitude. So if this cloud of points represents all of the observations of a single species in a single year, let's say 2010, and we know from our validation at the start that this species has a warm edge in the region. We took the 5th percentile of latitude here to be the warm edge, and then we did that every year. So this gave us a time series of edge positions for this subset of species that we knew had an edge in the region. But we wanted to do more than just get the edge dynamics. We also wanted to capture whether they seem to be correlated with temperature. And so we took the first few years of where the range edge was found and looked up what temperature on average the oceans were at that position along the coastline. And then we tracked that isotherm over time. So if at the start of the time series, the edge was found at 7.8 degrees Celsius mean annual sea surface temperature, we then plotted where along the latitude of this region, so how far north, we would expect this edge to be if it were tracking that 7.8 degrees Celsius. Here's what that looks like. The gray is the edge position. You can see it varied a lot, but it didn't actually move north, whereas the isotherm, the orange thing, moved pretty significantly north. So if this range edge were tracking mean annual sea surface temperature, we would have expected it to go from about 40 degrees latitude to about 42.5, and instead it actually didn't move at all. We did this for all the species for which we had edge data, which was 14 poleward and 29 equatorward edges. And we found that in both of those groups, about half of each group shifted north, but poleward edges both shifted further and had a stronger correlation with that isotherm than the equatorward edge group. The last result that I think is important to highlight from this paper is how stochastic these edges were over time. You might have already noticed that in the plot I showed you from the method slide, but all of the edges look like that. They had huge variation in between years. And we included this in the paper to make the point that a lot of studies on range edge dynamics have very few time points. And if we somewhat arbitrarily subset our data to have one old and one new time point, we could have misled ourselves, not just in the degree of change, but the sign. This is one poleward, one equatorward edge, just as examples here. Let's say you have an old year of data from 1972 and you revisited it in the 2000s with no data in the middle. Depending on which of these 3 years you did your resurvey, you might find no shift, a southward shift, or a northward shift. So I think it's important to really underscore how useful these long time series are to study something as stochastic as an edge. I told you that we tested the species interaction abiotic stress hypothesis in the previous paper, but we realized later that we really didn't quite do that because we didn't actually have a way to interpret the fact that species track temperature somewhat. So it wasn't that conclusive whether we saw that poleward edges tracked better than equatorward edges, but it wasn't that conclusive. Were they tracking temperature, yes or no? We decided to drill down on that question and incorporate a lot more data to try to get a big picture answer to the question, are marine range edges at a fine scale tracking temperature change? We had to incorporate multiple NOAA surveys for this project that use different survey methods. So to compare apples to apples, we used a species distribution model called VAST that basically converts the cloud of points that I showed you previously into a continuous distribution of biomass throughout the study region. So note, and this will become important later, that the way we measured an edge in this study was really different. It was a quantile of biomass as opposed to just presence points, which meant it was probably much more influenced by the abundance of the species. I'm happy to talk more about this model later if anyone's curious. We now have 3 regions, so we are already familiar with the Northeast, and we added in the West Coast and the Eastern Bering Sea. These dashed lines are not pirate treasure maps. They are the axis along which we measured edge shifts. That's because in some of these regions, expecting just north-south movement is not a great proxy for where you actually think the continental shelf species sampled by these surveys would shift, which is along the shelf. And so we instead, in the Northeast and the West Coast, used just a line along the coastline. The Eastern Bering Sea is a very complex coastline. There are islands. And so instead, we used a line just directly through what managers there call the middle domain, which is their productive sort of moderate depth shelf. these regions have very different histories of warming. That's probably why we picked them. The Northeast, I mentioned already, has had kind of consistent, almost monotonic warming. The orange line we already saw is mean annual sea surface temperature. In this study, we also, because we're really focusing on temperature tracking, we actually used summer and winter extreme monthly temperatures, so the hottest month in summer and the coldest month in winter, because we thought that more accurately captured what would actually be limiting a species range. They're probably not limited by means. You can see in the Northeast that basically all of these are trending upward and have since the start of the NOAA time series. Many people here are much more familiar with the history of sea surface temperature on the West Coast than I am. It has not warmed in a secular way, but there have been huge spikes associated with El Nino in summer, annual, and winter temperatures. And the Eastern Bering Sea kind of falls in the middle. It has warmed, but not linearly. It's warmed through these episodes of huge spikes in sea surface temperature that are associated with record low sea ice in the Arctic. So I mentioned that we used warm and cold temperature extremes here, so summer and winter. And we compared each range edge, whether it was poleward or equatorward, with each temperature extreme, summer and winter, which I tried to make a plot to summarize this, and I realized this plot from 1947 does it better. Intuitively, you might think, well, the poleward edge is the cold edge, so it should be limited by cold, and the equator edge is the warm edge, so it should be limited by summer temperatures. But in fact, depending on which life stage is most limited, whether it's survival or growth, each season could technically limit each range edge. So we compared, no matter whether it's a poleward or an equator range edge, we compared it to both summer and winter temperatures. I'm gonna walk you through the methods and then show you what we found. We did this with over 150 range edges from these three regions, which had anywhere from 20 something to 50 something years of data. First, we just quantified the edge over time. We've already seen plots like this from the previous study. Here is Atlantic cod, a popular species on this coast. You've probably heard of it. And over the time series that we have, our model found that the equatorward edge of cod has retracted very far north. That seems right. We know that cod populations collapsed and sort of retracted back into the deep waters in the Gulf of Maine. And this y-axis is the same as that dashed line in the map. So this is just showing it going up the coast. The 0 is the origin point for the Northeast, that's Cape Hatteras. And then once again, we asked what temperatures were found at the position of the range edge every year. And now we're looking at summer and winter temperatures at the range edge every year. So for this dot in 1993, that was about 750 kilometers up the coastline, what was the temperature? If a species were, if a range edge were limited by temperature, you would expect at least one of these lines to be flat because it would be stuck at the same temperature. It wouldn't really be able to persist beyond it, and it would be, it would look like a true limit. It doesn't have to be both because winter and summer don't need to be limiting for the range edge to be temperature limited. Here, it kind of looks like cod has maintained the same cold temperature. So this is the equatorward edge, so that kind of suggests that it needs to be at some level of winter temperature, and when it gets too hot, it has moved to stay in those winter temperatures. But in doing that, it actually ended up, it looks like, someplace with colder summers than where it started out. So it shifted so far north, it actually is someplace with colder summers than where it started. That's, I think, intuitive from this plot, but we also needed to quantify that in some way, and we ended up fitting Bayesian linear models to each of these time series to get a posterior distribution for the slope. What we wanted to ask is the slope of this line 0? If the slope of either line is 0, we can say this edge is tracking temperature and consistent with the thermal niche hypothesis. For cod, this kind of bears out our intuition. The red line, summer temperatures, have a negative slope, so they got colder over the course of a time series, but the blue line has a credible interval that overlaps 0, which suggests that cold temperatures, winter temperatures for cod stayed consistent over time. Like I said, we did this for over 150 range edges across all the regions. And I'll start out by showing you the results from the Northeast. The top panel shows the equatorward edges, and the y-axis is showing you which of those lines that we just looked at, winter or summer temperature, were flat over time. Some had neither line flat, so they ended up in just a totally different climate than where they started. Some had a flat summer temperature, winter, or both. And the x-axis is showing you how far the edge shifted in space. So how far up and down the coast it went. If it went down the coast, it's a negative number. The first thing you might notice is that I just told you in the Northeast, poleward edges have shifted further than equatorward edges. And looking at this, you would see the exact opposite pattern. Equatorward edges shifted north, and poleward edges basically didn't shift at all. And if they shifted, they were just as likely to go south. We were also surprised by this result, and I think it has to do with the different methods that we use to measure edges and the fact that this one is based on biomass, not just presence. We also found that a lot of edges here didn't track temperature, even though this is the region where we have the longest time series, the most consistent warming, where we kind of thought if species were tracking temperature, it would be most likely to be borne out here. We actually had a number of equatorward edges that overshot temperature. So they shifted really far north, which is a range retraction. Again, maybe related to abundance dynamics. The results for the other regions are easier to show you because they basically all tracked both winter and summer temperature. I want to be clear though that if you think back to the temperature time series for the West Coast and the Eastern Bering Sea that were so stochastic over time, this isn't so much a very strong proof that edges are tracking temperature change as it is evidence that we couldn't reject the possibility that summer and winter temperatures stayed the same where these range edges were because there wasn't a directional signal. You can also see the same thing for the Eastern Bering Sea. Almost all these dots are in the both category, meaning that it looks like everything is tracking temperature really well, but I think given that in the Northeast we had the worst temperature tracking, it mostly underscores that edges are very variable, temperature is very variable, and so it's hard to tease apart how closely they're really linked until you have a situation like the Northeast where the temperature signal is much less variable and you can really see how closely species are tracking it. Like I said, I think the data that we have to use and the way we measure edges really matters in understanding the dynamics of range edges. I think the jury is still out on whether poleward and equatorward edges in the oceans are systematically responding to temperature change differently. We did a lot of work on this, but there really haven't been that many studies using big data sets like the ones we did. And I think there's a lot more data out there that we could leverage to try to do more universal tests of this question. And it looks like edges are maintaining their edge thermal niche, but I think some of that is because we haven't yet been able to tease apart how closely they really adhere to temperature when warming becomes really consistent. That said, our results definitely show that there is a strong relationship between where edges are found and consistent summer and winter temperatures. So we talked about temperature extremes. I also promised that I'd talk about oxygen, which I don't have results to show you because this project isn't much earlier stages, but in the oceans, dissolved oxygen co-varies with temperature, but it's been proposed that what looks like temperature limitation of species distributions in the oceans is actually temperature dependent hypoxia. So it's not that species have a physiological limit related to temperature, it's that they have a physiological limit related to how much oxygen they can take in to fulfill their metabolic demands at a given temperature. This might seem like an academic difference, but oxygen and temperature don't always co-vary. So if a species were tracking one or the other, that would actually be very helpful to know for prediction. And I'm working with some oceanographers at the University of Washington to use the same survey data to try to tease this apart. It's challenging because there's not as much oxygen data as there is temperature data, but we're making some progress. So we're now going to talk a little bit more about dispersal and population dynamics. One of the exciting things I'm working on related to dispersal is also an EDGE project with Mary McElroy at UCSB, who's using eDNA to survey intertidal and subtidal biodiversity in California, which we're hoping will reveal the extent to which point conception acts as a biogeographic boundary by limiting dispersal versus by selection on individuals after they disperse across the boundary. So that will hopefully give us some insight into how much marine range edges are limited by dispersal around biogeographic boundaries. But where we really want to go with all this work, trying to understand what's driving the distributions of species in the Anthropocene, is prediction. It's really interesting to look back and try to think about why things happen, but our goal is to inform how we manage the future. So to move toward prediction, the universal tool in biogeography has been species distribution modeling. Most commonly something called ecological niche modeling, which basically works like this really great plot from a paper about 10 years ago. You start with the distribution of a species, just like the survey data I've been showing you. That's here. And then you identify some candidate covariates. They're usually environmental factors, although they could be something like the presence of another species. And you also gather data on those covariates. Then you can use them to map niche space for the species, which basically says you look up all of the values of your explanatory variables, rainfall, roughness, and southness in this model, all of the variables where the species was found. And you use that to construct kind of an envelope within which you think is suitable habitat for the species. You can then project that back onto a landscape. You can use it just to estimate a species range or to make future projections if you incorporate climate models. This approach doesn't tell you anything about why we think that the explanatory variables, things like roughness, southness, rainfall, would be driving the species distribution. So the mechanisms are all implicit. It's just kind of not a black box because we know how they work, but there's nothing describing the functional relationship between the explanatory variable and the response variable. We start out with some survey data and we end up with a forecast. But we don't totally know why in terms of what changed in a species' fitness, its abundance, its dispersal ability. So these models aren't necessarily the best tool for when those relationships between the environment and climate and a species distribution may change. And that could be the case for species that are not in equilibrium with the environment, which is basically all of them now. For species with major fluctuations in their abundance, which again is common, especially in marine species that experience harvest from fisheries like the ones I study. And when species are experiencing climates that they've never seen before, which sadly is also common. A way forward that's been proposed is process-based modeling, which makes these mechanisms explicit by actually linking the drivers and the response, which is in my case, the species distribution, via sets of equations, essentially. The ones that I'm going to be talking about today, the processes that will incorporate are dispersal, demography, physiology, literally what's on this plot, actually temperature-dependent population growth, even though I did not make the plot, and the environment. We're not going to talk about species interactions and evolution, although still very cool. There's a reason that more people haven't applied process-based models in biogeography. They're very data hungry, and it's difficult to estimate the parameters that you would need to make these explicit mechanistic connections. In addition, when you try to make process-based models spatial, and let me say, even if you haven't heard the term process-based models, as an ecologist, you definitely know what they are. Population models are process-based, dynamic energy budget models, climate models, but they haven't been that common in biogeography. And I think that's because it's hard to estimate these processes at the appropriate scales to start saying how things vary through a landscape. So what I'm working on in my postdoc is called a dynamic range model. It's a type of process-based model, starts out with exactly the kind of data we've been talking about, survey data on distribution and abundance, and you end up with a forecast of the species distribution, also in terms of abundance. But rather than keep all these mechanisms implicit, we set up a system of equations that we think describes the way that these populations work. They grow, they reproduce, they migrate, they die. And we fit that model to the survey data to estimate the key parameters. With that parameterized model, we can then make simulations of the future, and that's how we get our forecast. So it's the same model in both of these steps. And I'll walk you through a little bit of how it works. I don't have results to show you yet because we're still building it, but I'm very excited about it. Some of the reasons it's really exciting is that, first of all, we get these parameter estimates that are really interesting, estimates of things like growth and dispersal. It could capture transient population dynamics, again, really important in the marine realm where you have boom bust populations and huge variation in abundance. because we're trying to describe these fundamental relationships between the environment and life history parameters, hopefully this can project into no analog climates on which we don't have historical data. And we hope that all those things will make these models more accurate at short timescales, which is when we most need prediction for management. And well, I'll mention this in a second. So what I'm doing is a short-term forecast every year for a decade. In practice, that means that we take the historical time series, we reserve the most recent decade as testing data, and train the model up to about 2008. Then we'll compare that 10 years of forecasting to traditional species distribution models like the ecological niche model I just showed you. And this is a theoretical ecology project, but it's also a partnership with managers who are looking for ways to get an early warning of when species will cross management boundaries. This is a huge issue on the East Coast, where you have 3 fisheries management councils up and down the East Coast, as well as a lot of fisheries that are managed by the states. There are a lot of states on the East Coast, and they're very worried about species shifting across management boundaries. It's already happening and causing a lot of challenges. So one of the co-PIs on this project, in addition to Malin Pinsky, my postdoc supervisor, is Brandon Muffley, who's a staff member at the Mid-Atlantic Fisheries Management Council who works on ecosystems. We picked 4 species to start with in consultation with the fishing industry and managers in the region that represent both a variety of life histories. Some grow slowly, some grow very quickly, but also a variety of management challenges. Some of them we just don't know very much about their distributions and abundances. They don't have stock assessments. Others are very well assessed, but there's a lot of concern about them moving between regions. So I'll show you how this model is set up. At its core, it's a population dynamic model. So we have 3 life stages. There's no reason there couldn't be more, but we thought this was sort of a good compromise between complexity and tractability. Small juveniles, large juveniles, and adults. Each one has their own mortality rate. There's also a transition rate between the 2 smaller stages and the next one. And then adults produce offspring that become small juveniles. I promised you that this was a spatial model. And so this happens in a kind of cartoon approximation of the coastline in different patches. At the moment, a patch is 1 degree of latitude. So we've split up the coast into these 1 degree bands. And we have this process simulated in each of them. Again, fitted to data on what's actually in those 1 degree latitude bands from the NOAA surveys. And that means we also have exchange of individuals between the patches, either via adult migration or dispersal of small juveniles. You may also have noticed there's nothing in here that's climate related yet. We incorporate temperature by making some of these rates temperature dependent. Both for biological reasons and modeling reasons, they're not all temperature dependent. It's either the transition rate between life stages or the rate of offspring production by adults. So once again, we'll start with the survey data and we'll fit this simulation model along these patches with individuals from the survey sorted into different life stages and estimate the key parameters that were the orange arrows in the previous slide. What this actually looks like, these are figures from a paper in prep by a former postdoc in the Pinsky lab who's now faculty at York University, Jude Kong. And these are not real data, so I'm not actually showing you anything about the 4 focal species that we're studying. But what we hope it will look like is Jude's simulated data, where he fit the model to real data, although in this case it was simulated data itself, but we're calling it the observed data. And then we project into the future and hope that we can reconstruct key dynamics, which is to say the abundance of these 3 life stages across all the patches over time. So we fit the model, then we make these simulations into the future where we no longer have data, we just have the temperature. And hopefully we get a forecast that looks something like the real thing. And we'll do that again for every life stage, small juveniles, large juveniles, and adults at each patch. Our hope is that in the 1 to 10 year timescale, we will do a better job of capturing key features of range dynamics like range size change, range edge position relative to key management boundaries. And we'll capture those better than a statistical species distribution model. So my next steps on this project are to finish the model coding and fit observation models for these species. That's been non-trivial because some of the species in the survey are sometimes caught, they're often caught 0 times, sometimes it's 1, 2, or 3, but then sometimes it's also 8,000, 9,000, or 10,000 because they school. And so fitting an observation model to that data distribution has been a challenge. Once all those tasks are finished, we'll run these forecasts and compete them against other models. In the future, my next step is to incorporate the effect of fishing. You remember those orange arrows that represented mortality? We'd like to partition them into natural mortality versus fishing mortality, which of course varies over space and life stage, to understand how that affects the distributions of these species and hopefully have it improve our short-term forecasts and our ability to inform management. So I want to come back to this question I started with: why are species found where they are and not elsewhere, and what makes them move? Let me summarize the approach I've taken to this question to hopefully shed light on Anthropocene range dynamics in the context of climate change, and particularly in the oceans. I think we started with, and it's important to start this way with most of these questions, by thinking about what processes might be driving ranges. This doesn't have to be a perfectly constrained list, it won't be, but there are so many possible explanatory variables that we need to use our ecological knowledge to constrain them a little bit so that analyses can have tighter relationships between the hypotheses and the data. But with those candidate drivers, we can then build hypotheses about what might be driving ranges with certain traits or particular range edges or dynamics in a given region, and then conduct empirical analyses that actually test what is happening with ranges, or in the case of my work, range edges. This, in addition to yielding insight into those relationships, can demonstrate what real rates of change are and can showcase ecological surprises, like the few figures from other people's work that I showed you earlier in the talk, where species are not behaving the way we originally thought they would. That can tell us a lot about what processes actually matter. From there, I think process-based models are incredibly informative to explore future scenarios. We know there are limitations to using correlations of what happened in the past, and building simulations based on population dynamics and biogeographical processes that we suspect are really significant can help explore the space of future possibilities and anticipate how species will respond to future conditions in non-equilibrium states. Going forward, where I hope my research will end up is generalizing these ideas to understand the predictability of species ranges and biogeography across many species. Can we generalize process-based models based on traits? Is it possible to forecast ecological surprises? And how intrinsically predictable are species ranges in such a dynamic world? The end goal of all of this work is to provide more effective tools for species in the move. The biosphere of the future will not look like the one that we studied as ecologists early in our careers and we saw in the field. But I do think predicting future states of highly dynamic ecosystems is what will give us the best chance to protect them for the future. I want to end by specifically thanking the groups at UCSB that were a huge part in my education: Bren, NC's Ecodata Science, the Gaines and Halpern Labs, and my funding agency, the Linfest Ocean Program, as well as all of the co-authors listed here on the left. Thank you so much.
That was really amazing. Thank you so much, Alexa. If anybody has any questions, I'm sure that a lot of folks do, you can either type them into the chat and I can read them out to Alexa, or you can use the raise hand function if you want to ask it yourself. Looks like we have about 15 minutes or so for questions if folks want to stick around for this. So let's see. Got one question from Scott. All right.
Alexa, thank you very much. Very nice talk. My question had to deal with your process-based model for short time scales. And so a key element there is movement, is fish movement. And so I know Sea Grant studies off Cape Hatteras, as that thermal front moves north and south, have followed fish movement following thermal fronts over short time periods. So I was wondering if you could talk more about your process-based model and the degree to which it incorporates fish movement.
Thank you. I would love to. Let me go back to the slide that has the model structure, because I think that is a good thing to be looking at while we talk about this. So there's movement between the patches in this simulation model in two ways. Small juveniles can be exchanged, which they're not in the rates that we're modeling, they're not technically larvae, but it's what we use to approximate larval dispersal, or adults can move between patches. Most species predominantly use one of these pathways or another to disperse. So we do have some ability in here for adults to be exchanged between patches. The rate at which that happens, I think so far is not temperature dependent, although we can certainly think about making it temperature dependent. And in terms of the sort of fine scale temperature changes around Cape Hatteras, at the moment we're using in situ data from the trawl to fit these curves. So we have a very clear sense of what temperature the fish were actually found at. For the future forecast, we'll probably use ROM simulation that will again, give us hopefully a fine scale picture of what's happening with temperature. They can't always forecast kind of stochastic temperature events, but it'll be interesting to see in years where we have a rapid change in temperature, how well this simulation actually captures it for adult movement.
Okay, thank you.
I have a question for also just, I had forgotten to mention this. Thank you, Jen, for using the Q&A box. You're also welcome to put your question in there. So we have a question from Jen Cassell, who says, thanks to Alexa for the nice talk. One thing you did not mention is depth for the marine systems. One solution is going deeper to stay cooler. Is that in the NOAA data set?
Great question. It's totally in there. They record depth of all the toes. We did test for changes in depth in the Northeast study, the first study I showed you, to ask either, do species shift latitudinally or shift deeper? Or are there species that do both or neither? We actually didn't find any correlation kind of in any direction. The depth changes seem to be somewhat random. I still think that that probably is a big part of the picture. And it's something that we're unpacking a little bit more in the hypoxia work, because oxygen concentrations vary so much with depth. It's not part of the process-based model right now. If we find that the model does a terrible job of explaining species distributions and we think depth dynamics to, as you said, staying cooler waters are a big part of it, then we'll try to incorporate it. But the management interest is really in the geographical extent. So that's where we've been focusing.
Thanks so much. Do you have any other questions for Alexa? Okay. Oh, we got one more. Oh, no, I lied. That's just Jen still. Sorry, Jen. Thank you so much for knowing how to use the Q&A box, and I do not. I'm getting there. Awesome. Okay, so it looks like that is it for questions. You're also more than welcome to get in touch with Dr. Fredston over her email. And I know she would be more than happy to talk about her work with any of you. Thank you all so much for coming to this Monday's seminar series. Thank you so much to Dr. Fredston for being our guest speaker today. And yeah, we'll see you all next week.
Thanks, everyone. Take care.